Okay, so for the remaining of the day, what I would like to discuss with you is the following. So, we are entering into a period of synthetic biology where we have all these possibilities of logic gates, massive, reprogramming of biological systems, blah, blah, blah. And to do that, the problem or the challenge that we have is not to have a tool for making a short segment of DNA. The problem is when you have to make a massive reprogramming of an existing biological system. And therefore, that brings to the problem that is connected to circuit development, but it's the more practical angle of that, and is how to make genome scale engineering. I mean, not just to modify genes here and there. I mean, how really can you change many, many things in a biological system? So this is the slide that I shared with you when we discuss editing. And editing is insertions, deletions, type of replacements, but you can go further than that. And then editing is not only amending sentences here or there. You can do other interesting things. You can reconnect the text in a different order, okay? Or you can rewrite the story altogether. So I don't know if you are familiar with this novel that's called Rayuela from this writer. That is really, really interesting concept in literature. And it is, Rayuela is this kids game that you jump from one place to the other, like this, or like this, or like this, right? So in this book, it's a book, but depending on the order that you read the different chapters, you get a completely different story. Okay, so it's a very, very kind of inspiring approach, because that means that you can use the basic blocks for construction of a narrative, and then the narrative changes depending on the order. Well, the same happens with biological systems, okay? So editing may mean that you change some sentences here and there, that you reconnect the text in a different order, but you can even have a completely different story depending on the order and the structure of what you have there. So what redesigning a whole genome might be good for, okay? So we have been playing about these oscillators and these push on off counters and all these things, but are these games, or we are talking about more some things that are more serious, and can have more interesting biotechnological properties and applications? Well, the answer is yes, and I will show you a few cases. We're having some circuits that will control the levels of some metabolites and small molecules, and as a response of these levels, we'll be able to produce something that will be connected to that signal, for instance, a medicament or another active molecule. Then this is something that is already in the agenda of synthetic biology. So, well, for instance, even at the level of higher, say, organisms, chromosomal, big chromosomes and so on, it is possible, and has been done in some cases, to make small circuits that will, for instance, detect the presence of some, well introduced in viruses, some of these circuits, and then when the circuit detects some indicators of, for instance, cancer antigens, they will deliver some, say, response that will counteract the problem. Well, this is something that is very incipient, but is a very promising way to go. A more low-hanging fruit is to have bacteria that once it is introduced in the intestinal gut, in the gut, then they sense some changes in pH, for instance, and then they can convert that differences in pH in a response that will be beneficial to counteract the problem, right? So, for instance, if you have a high pH, then you may have some acidity problem in your stomach, and then you may introduce a circuit that will detect this problem and will produce a drug that might counteract the problem here, okay? So that's one of the main applications, but not only in health systems and not only in, say, health-related issues, but also, for instance, in plants. So in plants, for instance, you may have a situation, through which, depending on different inputs, then you may want to have different outputs. For instance, you may have water, age, and cold temperature, and depending on the combination of these inputs, you might be interested, or the plant might be interested, to produce something that will counter a drought, or that will produce an enzyme that will, for instance, avoid the action of insects. So, and again, using the same type of approaches that I discussed for the cello system, then at the end it all boils down to having a DNA sequence where you have all these interactions being programmed by changing the DNA. One of the most, attractive possibilities at the moment, in fact, is to use bacteria, to program bacteria to become as a kind of genetically engineered probiotics in such a way that they can really deliver activities in the intestinal lumen, and then they can change not only the composition of the microbiome, but also to have some beneficial activity on the cells that are there. Okay, so and in this case, for instance, you see that the logic gates here are implemented with quorum sensing, also with transcriptional factors, and also with these recombinases that flip around and give you some types of logic that will enable you to produce or not produce compounds of interest that will be beneficial for the host. But when you do that, we are not talking about making 1 gate or 2 gates or 3 gates. I mean, you are talking about making an immense number of gates that will be necessary to really have a real effect, right? So in this case, you may run out of promoters, you may run out of regulatory assets and so on, and you need to go at a big scale. So for instance, it has been calculated that for some bacteria that could be useful for therapeutical applications, you would require to make 1,500 constructs just with transcriptional factors. We would have to make in the range of 1,200 constructs with NOR gates, a large number of repressilators, a large number of toggle switches, and a large number of NOR gates. Remember I told you that the NOR gates are the basic unit for making complex circuits, okay? So, I mean, if you have to enter all these changes in one circular bacterium, forget it. I mean, it would be virtually impossible to go one by one and to implement this one by one. Well, furthermore, from a technical point of view, I mean, if you want to do this manually, by making classical cloning and so on, I mean, the number of PCR steps and cloning steps that you have to produce to really come to the point that you have an useful, say, bacterium should be astronomical. I mean, there's no way you can make 20,000 PCRs, clonings, etc. So I think that this is something that is completely out of reach. Same here. Okay, so then the idea is therefore that you have to develop technologies that enable us to enter many changes in many different types of genomes, and therefore here comes the big field of whole genome engineering that for the most part has to do with the capacity not only of synthesizing DNA but also to make sense of the assembly methods and the relations between the different compositions that we have in the system. This involves like 2 aspects. One of them is in silico. I mean, and this is now a must before you get committed into doing the right experiments and to start putting together different components for assembling big, say, segments. First in silico, it is possible to, growingly possible to have a model of what will be the result of writing a large segment of DNA and make a prediction of what may happen, right? So you can, well, compile some information. You have to run some model to see what will be the result, and then you have some, say, tools in vivo and in vitro to really implement that in a material fashion. So in reality, the ambition to have entirely synthetic genomes or heavily edited genomes comes back in history, even before the discovery of DNA as such, and some primitive methods. But as you may see here, it has accelerated in the last decade to the point that only in year 2010, it was the kind of milestone that was the first time that an entirely synthetic and complete genome was synthesized in the laboratory of Craig Venter. Okay, I will return to that in a moment. So at the end, what we would like to have is a situation in which we have a genome that can be either entirely synthesized from first principle or is redesigned from an existing one, in which we just go one by one through every component of the genome there, and then try to reshape and to re-edit the connections, the regulatory connections, in such a way that we produce a new type of bacterium that is synthetic. You may call it synthetic totally or partially, but certainly that it composed of parts that are rearranged, are standardized, and if possible are also orthogonal. So well, the idea here would be to to combine the best of the existing genomes with the best of the synthetic genomes, and then perhaps making hybrids between one or the other, so at the end we have optimal, say, chassis with various things. In fact, there is this tendency to keep the chassis part more or less intact, and then to maintain a domain of the chromosome as the part when you rewrite all these new features that you can enter in the genome, like logic gates, new circuits, and all the rest of it. So to do that, we have two strategies. The strategy that you may call top-down, or the strategy that you can go bottom-up. Okay? So in the top-down strategy, what you start is with a surrogate host that in many cases is yeast, and then you assemble the bits and pieces of the new genome, and then you deliver at the end the modified genome to a recipient. Or what you do is that you start with an extant genome, and then you reduce the composition of the genome, and at the end the results are basically the same. So well, the two possibilities are there and are compatible, preferably compatible, with advantages and disadvantages. And the main advantage of starting with something that exists and put things in and out is that you start with something that works, and then it's a question of say, securing that the new modifications that one enters in the genome are viable. You have with something of de novo, you are never sure that what you enter will work, and therefore you have to enter also some steps for selection. Let's discuss a bit in detail either of these two approaches, the bottom-up and the top-down. So the approach A would be using an existing genome as the starting point for designer genomes. Okay, so in that case, there are various possibilities to enter and excise segments of the DNA of the chromosome of the bacterium or the biological system that you start with. One of them, for instance, can be based on integrases. Remember that in the first part of the lecture today, I just told you that integrases are enzymes that are able to either integrate or to excise circular segments of DNA into a target by means of a small homology between the two. Okay, so this is something that, well, one tool that has been used extensively for entering big chunks of DNA into an existing genome is using these integrases. The other possibility is to use a recombination, and this is something that I also discussed in one of my earlier presentations. So if you want to deliver a double-stranded DNA cassette with an insertion into a given location of the genome, and you have enough sequences, enough homology in the flanking regions, then you can make a swap between the donor DNA and the target domain that you have there, and at the end you replace this DNA, this target DNA, by what you had at the beginning. Again, at the end you have a modified genome. You can have small segments of DNA, you can have big chunks of DNA that you can combine one way or the other. So along the same, this would be like more specific recombination, but you can also play with a more extensive recombination at the sites, and you can use homologous recombination for making this type of operations. Okay? Another tool that we also discussed in previous days is recombinering. So recombinering is this a technique that enables you to use small mutagenic oligonucleotides to enter specific modifications in given locations of a target chromosome. So you need a single-stranded DNA binding protein that comes in various flavors, but then the most important is that you enable a mutagenic oligo to invade the replication fork, and therefore through subsequent rounds of replication, then the mutation will be inherited by the cells. Okay, this is something that we have also mentioned before, and I will not return to that before. So, by making these site-specific say changes, remember also that you can generate chassis and different bacteria where you modify one of the triplets of the whole chromosome, or one or more of the triplets, and liberate some, say, meanings for some of the triplets for other things that you might be interested. Okay, so and remember that if you remember that there are 3 stop codons, and there have been cases where one of the stop codons have been reassigned to signify other things, not necessarily the stop codon. So for doing that, you need to combine, or up to very recently, it was necessary to combine. To go one by one, to divide the chromosome in a large number of segments and go one by one and enter the different mutations that are necessary in each of the segments, and then at the end combine all the segments in a single piece. So this is something that has been quite successful. So this is one possibility, and you start with something that exists, you insert big chunks in and out, or you just target the specific genes and produce allelic variants that will be more adequate for the type of programming that we are interested. Also, in the idea of this approach of top-down, then one possibility would be to make some type of genome reduction. Okay, so typical genome reduction is this concept where you have an existing genome, and then you go through the composition of the genome, and you decide what segments of DNA you want to change, what you want to delete, and what you want to keep. Okay, and therefore this is an exercise that first has to be made in silico, so you identify what path you want to take and see then what happens. So if you want to delete specific segments in the genome after you make this analysis, then you can use some of the methods that you have seen in previous classes that are based on homologous recombination. And if you remember some of the slides, what you do is that you generate a cointegrate between a donor plasmid and then the chromosome, and in the donor plasmid what you have done has been to create a segment of DNA that has homology to the ends of the DNA segment that you want to delete. And then by making first a cointegrate and then by solving the cointegration, then at the end you may delete an entire chunk of DNA that is in between. Okay, we have seen these techniques again in the past, and I will not repeat them, but basically this is one of the more, say, traditional and uncomplicated methods that you have to enter this type of big deletions or big insertions in the chromosome of a pre-existing bacterium or a pre-existing biological system that you want to modify. And by doing that, also you remember the story that I told you a few days ago, then you can get many different deletions in the genome of your favorite bacterium, in this case in Pseudomonas fetida. It has been made massively in E. coli, and it has been made massively in Bacillus subtilis, for instance. And then at the end you may delete around 5% of the whole genomic component, of the whole genomic contents. And then what you have here is basically things that you are not interested for, the programming and the behavior and the activities that you want to get at the end after the modifications. Picture that you have seen with the naked strain or this heavily modified strain that has been kind of pre-programmed to be a weak host for further manipulations. But this is if you want to delete big fragments of DNA in a directed way. But there are other ways to reduce the bacterial genome that are not directed but are random. So how does it work? Well, sometimes in a bacterial genome you have segments that are adjacent and that may not be necessary for given applications, and then in these cases, but you know exactly what they are, right? So what you can do, and we have done, and we and others have done, is to use transposons. So the transposons will have been engineered in such a way that inside the sequence of the transposon you have a site-specific recombination sequence, okay? So if you have two transposons, and one transposon gets here and the other gets here, and then you express in a conditional fashion a site-specific recombinase, then the two transposons, I mean the site for the recombinase here and the site for the recombinase there will recognize each other, and all this segment will be deleted, okay? So in that way you can make a completely random, say, elimination of big chromosomal chunks, and then that will be indicative that that part of the chromosomal structure is not necessary and you can get rid of it. So when you have this deletion, you can make another round of transposition, again another deletion, and another deletion, another deletion, and at the end finish with a bacterial genome that is reduced of many of the genes that are not necessary for growth under particular conditions. So this is what we did a time ago. So what you have is these transposons that contain inside the transposons these RT sequences that are recognized by site-specific recombinases. So the idea is that you make a round of transposition with this transposon that can be selected for one antibiotic resistance, and then you take the pool and make another transposition, and then what you have is a collection of strains with two transposon insertions in a randomly distributed fashion that have in between a segment of DNA that happens to be between the two transposon insertions. That if you now express this site-specific recombinase, then the region of DNA between the two insertions will be deleted, okay? And as this will be an entirely random deletion process with many different parts of the DNA being deleted and with different distances and everything, then this is a way to identify regions that are not necessary for growth. So, well, this is just a detail of the experiment, not really very interesting, but it's just to show you that indeed you can have insertions of the two transposons and the intervening region can be deleted if you express the corresponding site-specific recombinase.
How do we detect when we have got the least size of the gene?
Okay, well, there's a trick here, a detail that I did not mention, but I will, as you ask, I will mention, and is that inside the one of the transposons, we have a gene called PIRF that gives sensitivity to an antibiotic, okay? So that means that if the two transposons go to different locations, then in principle the strain will be sensitive to this antibiotic. However, if you express the site-specific recombinase and you delete the intervening regions, the transposon has been engineered in such a way that this gene PRF will be eliminated as well, and therefore you have a direct selection for the deletion, okay? So this is one of the various tricks that you can use. Well, genetics is a lot of, say, mental exercise, you know, because you have all these little, say, components that you can play with, and then you have to be imaginative to solve the problem. And in this case, the way the problem was solved was by making a genetic system in which the loss of the DNA resulted in the gaining resistance to an antibiotic, okay? Okay, so these are some of the results, for instance, of some experiments. So in one experiment, for instance, in a single shot, 4% of the chromosome was deleted from here to here because we had, there were two insertions of the transposons here and there, and then this is a chunk that was deleted. There was another deletion here that was way smaller, and in other subsequent deletion, then we had a deletion here and there. So in this case, for instance, about 10% of the genome of P53 could be deleted, and at the end, the result was a strain that was smaller than the original strain. I mean, the genome was smaller than the original strain, and still it maintained basically all the characteristics that were beneficial of this strain. Okay, so that means that you know, in many cases the chromosomes are packed with junk that is not necessary, and thanks to the methods of synthetic biology, we can convert them in more predictable and more, say, reliable things. Okay, the other approach, and you know, the approach A, if you remember, bottom up, is that you start with a existing bacterium, and then you start deleting or start making modifications, starting with an existing bacterium. But you can go exactly the other way, and then start a complete chemical synthesis of a genome. Okay, so until 10 years ago or something, it was completely impossible. But now, different individuals, in particular the laboratory of Craig Venter, was able to develop an entire pipeline for synthesizing and assembling large segments of DNA that, at the end, when put in a host, they work. So it was a very impressive endeavor, a very impressive outcome. So what do? Well, you start with a natural cell, you sequence the DNA, and then you take a look to the cell, and then you decide, just by either by computational means or by intuition, what genes you should keep in a completely artificial genome, and what genes you can erase, right? Because some of them are more necessary than the other. And then once you have decided the sequence, then you go ahead and start, well, first of all, design or write the sequence of the whole genome in silico, and then little by little you start synthesizing bits and parts of DNA in a DNA synthesizer. Now here comes the second part, and is to assemble the different pieces. And the technology that has been more successful so far is the idea of using yeast as a kind of surrogate mother, to bring the segments of DNA into yeast, and then as yeast is so good at recombination, by having the synthesized sequences having some minor homology regions at the end of the corresponding fragments, you can put them in yeast, and the yeast will take care of assembling them little by little, and then at the end you have a completely synthetic genome that obviously will not work in yeast because all the entire gene expression machinery will be different, but you can isolate the whole genome from yeast, and then by using various tricks you can enter the synthetic gene, the synthetic chromosome, into a recipient cell to have a kind of transient situation when the native chromosome and the new chromosome coexist, but then you enter in the new chromosome some features that make that over time this old chromosome will be lost, and the only that will be maintained will be the synthetic one.
But how does the yeast know how to make? Because we are only putting the individual DNA pieces.
No, but in the genome what you have is a yeast origin of replication, just as a little, say, part in the whole genome, and this is the only thing that yeast can recognize in the whole, say, DNA sequence. But the DNA sequence is completely inert in yeast because there are no transcription factors. I mean, there is nothing in the yeast that can recognize the sequence as its own. So all that yeast makes in this case is to assemble, to provide the recombination machinery to assemble all the bits and parts of the that you provide to the yeast, to assemble them in a single circular piece. Okay, and then to put this plasmid, to put this, say, synthetic chromosome into that, then you have to encapsulate this into a vesicle that will be fused to the cell. I mean, it's not piece of cake. I mean, there are some tricks there, but eventually what you have is the possibility of assembling a complete genome. Obviously, the genome has to be small. I mean, if it is big, it will be a big problem. But then you can generate this as a completely synthetic thing, and then the synthetic thing goes into the cell, and then it displaces the original chromosome, and eventually the hardware of the cell works with the software that we have introduced from the outset. One of the, say, transcendental say, consequences of this type of experiment is that for the first time it proved rigorously this idea of bacteria with a software and a hardware, okay? Because it shows that you can maintain the hardware of a cell and replace the software altogether, the software that is encoded in the DNA molecule, and now the hardware will read and execute the instructions that come from the entirely synthetic piece of DNA. So I think that, I mean, conceptually it was a kind of interesting thing. Okay, so how do you go in detail in this thing? Well, you have to synthesize oligos, then the oligo has to be assembled, and at the end you make bigger and bigger and bigger piece of DNA. And as I just mentioned to your colleagues, so this piece of DNA go into yeast, and in many cases yeasts are able to really assemble a multitude of DNA pieces just by using small homologies that you engineer in the extreme of the DNA, right? So in this particular case, the case that was the first time that it was made, then the operators or the scientists here, what they did was to generate a large number of operons or big chunks of DNA, and they assembled them in a single circular DNA that then was passed to a donor cell, and then in this chromosome there were some antibiotic resistances or some features that could displace the chromosome of the donor cell and make sure that the offspring or the consequence or the outcome of this genome coexistence was just cells that had the synthetic DNA. There's a technical issue here that I want you to keep in mind, and is that to do that you need a tremendous muscle in massive DNA synthesis. And production of big chunks of DNA or big products of DNA has been one of the main breakthroughs. I mean, it may not look like a breakthrough because it's a piece of DNA, right? But technically, it has been the technology that has really enabled to think big and to think on this larger scale editing, larger scale design of chromosomes and biological systems. Well, the cost of synthetic DNA is simplified here, and at the beginning, it was very modest, but now, I mean, with modern technologies, you can really develop large segments of DNA that are reasonably, priced, and now, you can get big, segments at low prices. Well, this slide is a bit old, so now the prices have come down, and now any laboratory, I would say nearly any laboratory, can reorder large chunks of DNA, not for genome synthesis, because this is still a super big segment of DNA, but for daily operations. In many cases, it's better to spend the money in generating synthetic DNA pieces with all the compositions and all the parts and so on, rather than going through the pain or having to PCR up the different segments, put them together, make a ligation, and all these things, right? So, but one thing that I want you to keep in mind is that DNA synthesis is not error-free. And one of the main limitations today in DNA synthesis and engineering genomes of high quality is the fact that even the best systems for DNA synthesis commit mistakes. And this is something that in many cases is a pain in the neck, because you think that you have solved a problem, and then it turns out that at the end you get to a final construct and it doesn't work. And it doesn't work because the original sequences that you started with happen not to be completely correct. And you know that in a complex construct, it's enough that one gene mutates in one base to kill the whole thing. So in the last few years, there's this growing interest to develop new methods of DNA synthesis that can really deliver super high quality DNA, because otherwise it will be impossible to progress in the direction of making synthetic biology agents that can perform as we want. So for assembling large DNA fragments, well, there are various procedures. As I said, one of the best consists of just having everything assembled in yeasts, but in other cases there are other methods that people have developed for doing that. And some of the methods are, for those who doesn't like to use yeasts, then you have other choices, not as good as yeasts, but in many cases you can get away with it. So there are some methods, for instance, this sequence and ligation independent cloning, that, to be frank, has not been super successful, but at some point it was a kind of big promise. And the idea is to generate segments of DNA that have sufficiently long protruding 5' ends that can go into vectors and eventually into the chromosomes without necessity of ligation. Well, this had some limited success, but one of the most successful, new methods for cloning large DNA fragments in a multiple fashion is what has been called Gibson assembly, that has already more than 10 years. So the advantage of Gibson assembly is that it's a cloning method that does not depend on restriction enzymes. So it is based on creating some minor homologies at the end of the segments that you want to put together, and then with a exonuclease that is called T5, chew up a little bit the extremes in such a way that the overhanging genes get exposed, and then very easily they can anneal with other segments that keep at the ends matching sequences with the others. So you may have, I don't know, a large number of fragments put together, and you have a code that will segments to identify and to bind partners. Then at the end, in many cases, you can put together many partners at the same time. Well, in one of the cases, by using a combination of GIST, surrogate, recombination, and also this Gibson cloning, then these people, Gibson and Daniel Gibson, in the laboratory of Craig Venter, were able to, in a single shot, to put together all these little segments that you have here. Okay, I don't know how many, maybe there are like 100, 200 segments. Each segment covering, you see that each segment is more or less the same size. There's no biological criteria, it's just a purely physical criteria. And the idea was to generate one by one all of these segments here, then with this homology code at the end and everything, and then make a mix and put the whole mix in a super efficient in recombination GIST. And then at the end, it was possible to assemble an entire chromosome of this bacterium that is called Mycoplasma genitalium. So Mycoplasma is a bacterium that is in some of the species are pathogenic, but the advantage of this bacterium is that it has one of the smallest genomes known, and therefore it has only about 700 genes or something like that. And therefore, in terms of the synthesis, is way easier than E. coli or Pseudomonas that has 4 or 5 times more. So this Mycoplasma was one of the first cases where an entirely synthetic DNA segment could be a chromosome could be created, and obviously this made headlines all over the world, because it was argued that this was a created cell. If you like, creation is a very strong word. Creation is that you have nothing and you have something. You may discuss whether the term creation is a good term or not. I think it's more a PR thing than a reality, because there's no creation. In this case, has been a kind of leveraging the hardware of the cells to interpret the software that we have introduced. But anyway, so this paper made headlines time ago, and since then it has been a reference for further attempts that have followed to make entirely artificial chromosomes or entirely synthetic chromosomes for different purposes. In any case, I insist that to this day, GISTs are the favorite surrogate hosts for synthetic genes or for synthetic genomes, at least for genomes that are small, because the genomes can replicate in the bacterial host as if they were plasmids, because the yeast will not notice that it's a chromosome, it will just notice it's a piece of DNA, and as it is completely orthogonal in respect to the rest of the physiology and the genetics of the cells, then it will be there and will do nothing and will replicate. And once the synthetic genome is assembled, it can be put back into Mycoplasma or into the destination, strain, and then at the end it will do what we want it to do. So this would be the collection of fragments. So you first make some assemblies in vitro, you produce the fragments, and then eventually you make them all to be put together by the GIST recombination machinery. Okay, so there's this other, technical aspect that is quite interesting and is not yet completely solved, I have to say, so there's some room for improvement, and is how to make this genome transplantation. Okay, so for the time being, the best idea is to make a cell where you have a coexistence between the synthetic cell, the synthetic genome, and the existing genome. And then, in many cases, or at least the best idea that people have figured out, is that in the synthetic genome, you add in the genome some characteristics that, over time, will make the synthetic genome to displace the recipient genome. And for instance, antibiotic resistance or some metabolic capabilities that will make things to, will make the over subsequent divisions of the cells will be such that the existing genome will be diluted out, will be displaced, and then the new genome will take over. By the way, it's a historically interesting detail that when this paper was sent for publication, the most difficult part to convince the reviewers that this was authentic was to show that in fact the entire genome that was inherited by the synthetic cells or by the so-called created cells had nothing of the pre-existing genome here. So in other words, how do you know that the synthetic that the cells that you have at the end are nothing but the result of the instructions that you have at the beginning there? So it really requires some extra, to walk a few extra miles to show that that was the case, that you could really replace completely the instructions pre-existing in the genome that was before there, the replacement of this by the instructions of the synthetic one. Okay, so it was an interesting thing. But in any case, the kind of vision that some have for the future of genomes a la carte in the future is that you may have this type of interfaces. Well, these interfaces are a simulation, don't think that it exists, but you know, you say, well, what do you want? Well, I want a bacterium. What do you want? Gram-positive, gram-negative, whatever? Okay, so I want a gram-positive. Okay, good, great. So what type of division type do you want? Okay, I want septation. Perfect. So what is your favorite type of metabolism? Well, you know, it's gram-positive, but I prefer not to spend money in nutrients. Let's make it photosynthetic, okay? You click there. Okay, so what genes you are interested to have? You make a list and you click here and there. And then what type of control you want to have to make sure that it does not escape the laboratory? Okay, well, I make it auxotrophic for something, and then it will not be possible to proliferate without it. And then, okay, then you just press a button, and then after a while, you get this type of things. This is a kind of vision, okay? And I don't know to what extent it's realistic or not, but you know, this is something that is on top of the table, because at the end, one would like to have a universal chassis, a universal platform that will be able to produce all types of synthetic biology agents following the same pipeline, okay? Whether or not that's possible, we don't know yet, but you know, this will be like the grand vision of some synthetic biologists, in particular Craig Venter, that argues that once you have dominated and domesticated the entire pipeline for DNA synthesis, there's no limit to the programmability of the biological system that you can get through this approach there. But this is not the end of the story, because bacteria are bacteria, but obviously the ambition is not just to program bacteria for do something interesting, but to program entire organisms, right? So years ago, a new project started involving many different groups all over the world. It will say, well, let's try to synthesize entirely the genome of yeast. And yeast have 16 chromosomes, and then, well, it's not a piece of cake, it's a lot of DNA. So as we may not have a single laboratory the capacity to synthesize the whole thing just in one place, let's agree to make a division of the yeast chromosome of the yeast genome in 16 chromosomes, and then we make a meeting, and then we decide who is going to synthesize which chromosome. Okay, so the sequence was known, because the yeast genome was sequenced a long time ago, but the idea in this case was to make a kind of offer and to make a kind of contest and say, well, you know, who wants to synthesize chromosome 7? Okay, Edinburgh, in collaboration with BGI, that sort of thing. So there was a big consortium that is still going, and this is a slide that is for, it has already some years ago, but you see that some sequences are already completed, others are half-half, others are not yet completed. But having a yeast with a completely synthetic genome is coming, and it's coming very, very soon. And there's someone called Patrick Kai in Manchester that is now taking the lead of completing this whole thing and having a yeast that is completely programmable or has been already programmed on the basis of synthetic DNA. But this is not the end of the story, and obviously after yeasts, we have humans. And in recent years, there has been an initiative called GP Right. And GP Right is this concept or this endeavor or this ambition to have an entirely synthetic human DNA. So why? Because if you remember one of the slides in my first presentation, there's this concept that what I cannot create, I do not understand. Remember this blackboard with this sentence put there. Okay, so this is not yet finished at all, so this is incipient, and in fact the amount of money that is required for doing that is astronomical as well, and not everyone is convinced that by having a completely synthetic DNA, we would do many things with it, okay? So I don't know what is your thinking, maybe you have an opinion on that, but I also remember that when DNA sequencing technologies started to be popular, there was a big discussion on whether sequencing the entire human genome was a good idea or not. You know, people say, well, this is ridiculous, why you want to have these sequences? And then over time, it has been very clear that having the entire genome of humans has been a super important thing for medicine and for many applications. So now I see the same discussions with the complete synthesis of the human genome. And you know, people say, well, why do you want to have a synthetic genome? Well, you know, maybe we don't see the importance right now, but in the future, having a reference synthetic genome will be important. This is something that perhaps we can bring to our discussion on the last day, because it's important to have your feedback as well. What, you think it's a good idea to do this, or you think that is a waste of time because we still do not understand well how the human genome works? But anyway, it's something that is underway. And then just to finish the entire scenario, I also want to mention that the last few years has seen an immense interest to produce synthetic cells, not just to have a chromosome or to modify here and there, bottom up, top down. No. Let's start creating a bacterium cells from first principles. And then what synthetic bacterial cells might look like. Well, a synthetic bacterial cell, in principle, should have all the components of the gene expression flow. Obviously, there is some in silico design aspect here. You need to have some tools for for assembly, and then ideally, we would like, a synthetic bacterial cell will have to have all these components here. Can we make it simpler? Because obviously, despite the simplicity here, you still need many things and parameters to be fixed. People say, well, we can make it simpler. We can have a situation where we just have we have far less genes here, and then just we ensure a minimum level of replication in the DNA and a minimum level of transcription here and a minimal number of proteins that instead of generating, building blocks for metabolism, will import these building blocks from the outside. We will provide amino acid, we will produce nucleotides and so, and therefore many of the genes that will be necessary in a completely autonomous cell will not be necessary in this case. And some authors say, well, this is still very complicated. Let's try to do something like this, right? To try to make a self-replicating vesicle that is based only on RNA, and we provide the substrates for this type of replication. Okay, so you can go as simple as you want, and then the decision is where you put the limit or what is a life system and what is not a life system, you know, and this is again something perhaps for discussion. But you can really simplify the characteristics of life. You can really go back in evolution and think on a form of, well, I don't know, I would not call this a living entity, but certainly will be closer to the situation at the origin of life, where you have a self-replicating RNA that is also, embedded in a bilayer membrane, okay? And maybe this would be the very basic unit that perhaps can be the starting point for building a synthetic cell. So there is a considerable interest in the US and in Europe. I really don't know how things are here, to really make a kind of Manhattan project. I mean, not with the intensity of the first Manhattan project, but indeed to really be able to construct a completely synthetic cell, with the argument that constructing a synthetic cell will be the ultimate, the ultimate proof and data for understanding how life system works with all the consequences that may have. So different laboratories, as I said, in the US and in Europe. And in Europe, there's a super famous lady called Petra Schuile, that is a German scientist in Germany, that they are producing not synthetic cells, but cells that are endowed with some properties that are characteristics of life. For instance, she has been able to develop some vesicles that are able to divide and produce two daughter, say, vesicles, and she's been able to assemble some basic components of the cell division, say, machinery and so. So at the moment, the idea is to start with a vesicle and to put in the vesicle some bits and parts that are representing the basic functions of a cell, but still the time has not come to put everything together. So I don't think that we have a synthetic cell coming in the very near future, but it will come sooner or later. I mean, it's very clear that at some point we will be able to create something similar to what we call life in the laboratory, okay? And I don't think that is going to be a super big deal, but in any case, this is something that was considered to be a kind of holy grail of biology, but I think it's now something that is quite at hand. Okay, so this is what I wanted to tell you today, and first of all, about circuits, and well, you had a bunch of them. I hope that you at least remember the simplest of them, the necessity of logic gates, how you combine them, the recombinases, the possibility of making them with metabolites, but also with transcription factors, with column sensing, with multiple bacteria. I mean, you have to remember the details, but at least I want you to remember that you have many possibilities of implementing computation with biological things, whether the things are transcriptional factors or are entire cells, okay? And that's something that will for sure grow in the future and see how develop. The main bottleneck, in my opinion, and in my opinion of many others, is this aspect that we discussed yesterday or the day or last week of context dependency. Things don't work identically in all places. So depending when you put them, the temperature, the interaction, everything may be different. So this is still a big issue that requires to be solved before this computation that we all think that will be interesting can be real, okay? But in any case, we are in this super interesting stage of the development of the technology. Okay, so that's what I wanted to tell you, together with all these new possibilities of making DNA synthesis, and at the end even having synthetic cells and all these possibilities that are open for the future. Okay, so now it's time for a coffee, and then we'll return quickly for making a wrap-up to plan a bit the IGM thing, and then it's up to you to hang around or to go, because I think that today, given that your colleagues will not come here, it may make no sense to prolong the meeting here, but you are very welcome to stay if you want to stay the final hour. But now let's go for a coffee, okay? Expressing interest. So please, if you are, I don't want to twist your arm, okay? So if you don't want to participate, it's okay. So don't feel pressure. You know what I mean?
No, it's okay. I wanted to do.
Okay, great. But then please send me an email, because I have to organize this a little bit. So unfortunately, as we don't have everyone here, I really don't know whether we can make progress with organization, because two of you indicated that they were interested, but not as kind of single persons, but joining a team. So, but I really don't know what is the situation right now. Maybe tomorrow we have to discuss this a little bit.
Yeah, we have to discuss it about he's not here. He has a.
No, I know. But do you know, because the other day I noticed that there was a little group discussing there.
Yeah, but I don't think we formed teams at that point.
Okay, so it's not that there will be a team.
Yeah, but mostly I and Abid will be on team. That stuff, because we send the mails together.
Okay, very good. So please do send me these messages, those of you who have not sent, or tell your colleagues to send them to me, so we can reorganize, because there will be a large number of presentations and we have a limited time, and then it will be better to have it a bit organized. So for Wednesday, the idea would be because the plans for Wednesday are kind of evolving. So we will meet before lunch, and we will have this little discussion on art derivatives and societal aspects of well, I tell you, you're going to be shocked on Wednesday morning, okay? Because what I'm going to tell you is different, you know, of everything I've told you so far, and it will be a bit. I'm sure that you will remember that session, okay? Well, anyway, so then after that, I would, as I said, I would like to have a kind of feedback from you about the course, what you have found more interesting, and so on. So on. Then I would like to have the 2 grad students that are involved in the course to give us small presentations, and then finally we make the presentations of the IGEM, you know, and depending on the number of participants, we will give them more or less time. So that's also important. That's why we need to know exactly how many people will participate. Okay. Okay. Well, I don't have many more things to deal with today, so I don't know whether you have any other issue or anything, but you know, otherwise we're done for today. And I expected.
Yeah, anyone of you interested in forming teams?
Yeah, actually, I love her idea, but I don't have any teammates right.
Okay, well, I mean, so
I have ideas, but I need people from other domains to help with the execution.
Yeah, I can be in one of your teams.
Okay, so can you please get organized, and then the day before, tomorrow evening or Wednesday morning at the latest, you tell me whether the participants are going to make presentations individually or whether you are going to team up. In any case, teaming up, and then you make a team, whether you want everyone in the team to say a little bit of the project, or whether you want to elect a representative. Okay, I mean, the teams will be small, right? So you are 2, for instance. I don't know. Please decide whether you want to have your little few minutes and then the other few minutes, or you prefer to kind of delegate the whole project, presentation of the whole project in just one person. Please decide that in advance, right? Because as we will have many presentations, we should load the PowerPoints ahead of time.
Like you can decide how much time you are giving to each team, and then you can decide how they are going to.
Well, the thing is that I need to know how many teams and how many people will participate, just to just earmark some time during the day for the presentation. So we have a kind of fair time share for different projects. Okay? Yeah.
We'll discuss today and we'll let you know about the teams.
Sounds
I will let you know by tomorrow public, I guess.
okay. Sounds very good. Yeah. Perfect. Okay, so I think that that's basically it. I don't know if you have any other question or any other. Okay.
So we'll discuss our ideas also, and then if you need any help
Very good. So you are welcome to hang around and.
or sometime.
Sure. I mean, so you are left here to your own devices, so feel free, because we have the room reserved for until 6. So I think that you can hang around and discuss whatever you have to discuss. Okay, great. So if nothing else, I will return to my office and then I'll see you tomorrow. Okay? For the final talk that will be about metabolic engineering. And I hope that one. Well, I will have 2 parts. First will be metabolic engineering at a small scale, and then the last one will be metabolic engineering at a big scale, at a planet scale. So it will be fun, I hope.